Okay, good morning. Last day. Woo! Is everybody everybody excited? So this morning is going to be focused on PTMs, and we're going to start with a second talk from Tina, and she's going to lay the kind of groundwork in terms of what kind of information you have with respect to PTM peptides and what kind of information can be exploited, and then we'll have a talk from George afterwards. To talk in the more sort of formalized analysis, how the software is actually doing this, and then we'll have a tutorial later on to practice. So Tina, please.
Okay, so some technical issues. I tried to do it this way. So welcome also from my side to the last day. And yeah, as Ben already introduced, the topic of this morning will be post-translational modifications. And my pleasure is to do now a little bit more, again, the introduction, broad view on PTMs, and I will just give you maybe towards the end some very DIA-specific considerations, and then in the second presentation, George will take over and go a lot more into the software analysis part. Okay, so again, I've structured my presentation in 3 parts. We will start with the introduction into PTMs and talk then at the end a bit about the current technical challenges that we are facing. And then in the second part, I would like to focus on one of these challenges, which is the PTM site localization. And I know this came up already several times. So Alexei has shown some slides already, and also Mike has shown some slides. However, I think I will go a little bit more into detail what they did, and hopefully you will benefit and maybe understand the problems a bit more in detail. And then in the last part, as I said, some specific considerations for PTM in the context of SWATH and where we stand and where we might go. Okay, introduction into PTM. So what is a PTM? So after translation of mRNA by the ribosomes, polypeptide chains are not ready. Often they undergo various different kinds of post-translational modifications before they become the mature functional protein. And these PTMs are really very, very diverse. What most people understand when they think about PTMs is reversible addition of chemical groups to the N-terminus or the C-terminus or the side chains of amino acids, like phosphorylation, methylation, acetylation, oxidation, all these type of PTMs exist. But there are also another group of PTM where not just small chemical groups, but large proteins are attached to side chains or to termini of proteins, and this is examples for this are, for example, ubiquitinylation or simulation. Then we have also the reversible addition of other complex molecules than polypeptides, like sugars or lipids or ADP ribosylation, so other very large complex structures. And while all of those modifications here are reversible, there are also irreversible PTMs. Irreversible cleavage can occur. Typical example, proteolysis, so cleavage of the N-terminal signal peptides or C-terminal signal peptides. And there's also a process called intein splicing. That's a process that is similar to mRNA splicing, which takes place on the mRNA level with introns and exons, and the same can happen actually on protein level with inteins and exteins as well. And then there is also irreversible amino acid modifications, and one example is, for example, deamidation. So the purpose of this whole slide is to show you once more that PTMs are really very, very diverse. They lead to the diversification of the proteome, and as different as the chemical properties of these PTMs is as different is their function or their effect onto the proteins where they're attached to. So almost every protein is modified at least once or many times during its lifetime in the cell. And that's what we call this PTM code. So the summary of all the PTMs that can take place on a protein is what we call the PTM code. And this PTM code really regulates and fine-tunes the function of the protein in space and time. And these functions are, the typical functions that, pretty much all functions you can think of: localization, activation, deactivation, signal transmission, solubility, stability, degradation, all these things you can think of what a protein might be used for in the cell. And unraveling these PTM codes, so to understand which PTMs are happening, how these different PTMs on the same protein might communicate, how they cross-talk with each other, that's really one of the great challenges that molecular biology faces at the moment. And the fact that it is really a very hot topic, and it gets even more hot topic in the field, you can see from this figure where we plotted the number of PubMed publications that had the term PTM in the abstract or in the title, and you see that in the last 40 years it's almost exponentially increasing, and more people are interested in that. Okay, how many PTMs are there? So there is a useful database called Unimod, where all the PTMs, the described PTMs, are listed, and currently there are like 400 different biological PTMs which are known. And here are two figures from two publications where you see the frequency of PTM. So this is from SwissProt, and this is from a database called Phosphosite Plus, which contains PTM information for human, rat, and mouse, I think. And you can see that both of these figures show the frequency of how many PTMs of certain type are listed in these databases. And the winner is phosphorylation. So by far the most PTMs we find is phospho. Then we have maybe acetylation or ubiquitinylation, some methylation, and then very quickly it goes very down in the number or the frequency that we can find in human in that case. And however, what I think is very important to keep in mind when looking at such figures and interpreting such figures, you of course might think, okay, protein phosphorylation is really the most important PTM the cell has to regulate stuff. But yes, I think these numbers that we see here are, of course, and it might be true that this is the case. However, I think these numbers are also very biased by our ability to actually be able to detect these PTMs at all. So very biased by the analytics that we have to make them visible. And that's also a big problem or a big challenge for us to keep that in mind. Okay, so how can we measure now PTMs, and how can we measure proteins that carry different PTMs? And I think there are two main strategies what people do. So either they work with antibodies. So they work with antibodies that are specific for a certain PTM or even a certain PTM site on a protein, and then they do Western plots or Luminex assays or protein arrays. And these methods are very valid and very cool. They can be very sensitive. They can also be highly multiplexed, but they have all the big disadvantage. They rely on a good working antibody. And that's why I think the king of analysis method that most people use for PTMs, definitely the most used method in the field when you look at publications, and that's definitely mass spectrometry. And we can use mass spectrometry to analyze PTM carrying peptides because almost every PTM changes the mass of the peptides or the proteins that we're analyzing. Okay, so how does mass spectrometry and PTM come together? So what you see here is the typical PTM workflow that we use in bottom-up proteomics. And it's very similar, of course, to also the general PTM workflow. However, I would still like to quickly go now through these different steps and just give you some thoughts or some comments to these different steps. What's of course clear is that every single step that you see here is absolutely crucial and equally important to get a good result at the end, right? So none of these steps can be neglected. The first one is the step of protein extraction. And as in any proteomic experiment, first of all, you need to extract the proteins from cell or from some type of sample. And yeah, that's always a very important step. However, when it comes to PTM analysis, it's especially important because in comparison to the total concentration of a protein in the sample, which is, I would say, in the order of a few minutes rather stable, because before the total protein concentration really changes dramatically, you need to activate transcription, translation, and all that stuff. This is very different when it comes to PTMs, because PTMs can change very quickly. Many PTMs are regulated by an interplay of proteins that attach the PTM and remove it, like kinases and phosphatases, acetyltransferases, deacetylases. So big changes in PTMs can happen within seconds. And that's why when you perform protein extraction, the very, very important point to keep in mind is you must somehow maintain the PTM status on your protein. You must make sure that not during your extraction process, this PTM somehow changes. And what people do in the field to achieve exactly this, they work with buffers or in this lysis process that contain 8 molar urea, for example, to completely denature all protein activity. Also, 6 molar guanidinium chloride is often used, sodium deoxycholate, or this trifluoroethanol is also gains more and more attention to use this in this easy phos method that was published by the Mann group to maintain this PTM status. Other things to consider is PTM inhibitors, so to inactivate phosphatases, deacetylases to prevent the cleavage of the PTM. When you work with bacteria or yeast, it also can be very useful to work with TCA, trichloroacetic acid, because TCA can kill all enzyme activity very fast without lysing the cell. With human cell culture, you can unfortunately not use this. And then also people try to work always either very cold to prevent reactions ongoing, or they do just the opposite. They work very hot and inactivate enzyme. So all of these are things to keep in mind, to consider for a PTM extraction protocol, even more than for when you are just interested in looking at total protein contents. Okay, the next step is an optional step, protein enrichment. So you do not necessarily need to enrich for proteins. Still, it will help you in the sensitivity of your measurements. And typical enrichments, what people do is, for example, co-immunoprecipitation or affinity enrichments of specific bait and target proteins, which helps you to get rid of all the other proteins and just get your protein of interest. And then you can also look into PTMs of this protein of interest. Another typical enrichment on protein level is done in the field of histones, where people have developed protocols to specifically just enrich the histone proteins. Or there are other subproteome enrichment strategies, like using beads that bind all kinases, and then you have the kinome and stuff like this. The third step is proteolytic digestion. That's again an essential step when you want to do bottom-up. And again, this is, of course, a very important step also for global full proteome analysis, but it's especially important when it comes to PTMs, because we all know that when we work with our favorite enzyme trypsin, we usually are not able to cover the whole sequence of the protein. We will always have gaps where there are peptide areas which are invisible because they are too small, too big, or they are bad flyers. And if you're interested in a PTM that is exactly in that area, you're screwed, right? So with trypsin, then there is not much you can do. And that's why it actually makes, maybe in PTM analysis, a lot more sense to think about alternatives to trypsin. So just to give you one example, so let's assume you are here interested in PTM going on that tyrosine. If you work with trypsin, you unfortunately end up with a very small protein that you may not be able to identify or analyze. And when you work with enzymes like lyc, gluc, arc, asp-N, significantly larger proteins develop, and now your trypsin sits in other patches. And yes, there is a good reason why people like to work with trypsin, but still, as the other proteases often are not as efficient, as specific and stuff like that. But still, if you're interested in covering a complete protein sequence, strategies like this can help you. Okay, next step, PTM enrichment. Again, it's an optional step. You do not necessarily need to do that. But if you have a PTM enrichment strategy, it can definitely change the world for you. You can measure much more sensitive with a well-working enrichment strategy. And the typical PTM enrichment strategies people work with in the field that are established and that work very well are enrichments for ubiquitin by working with antibodies that recognize the K, the lysine GG sequence, and this GG comes from the ubiquitin, and when you work with trypsin, you will generate these KGG peptides, which you can enrich with specific antibodies. Of course, phosphorylation, we all know, titanium dioxide, iron, IMAC, you can use to enrich for those. Acetylation, again, often done with antibodies that recognize the acetyllysine peptides. However, there are definitely a lot of PTMs for which we so far have no enrichment strategy available. And that might be also one reason why they are all in the ground of frequency, how we can detect them. And developing these new strategies is definitely something with great potential, or where you will have a big impact. Peptide fractionation, the next optional step. Again, if you have a very, very well working enrichment or a good protein enrichment protocol, you might not even consider doing that, but it can also help you. And what people do here typically is an offline basic pH reverse phase fractionation, cation exchange, anion exchange, trimodal mixed phase columns, everything that people also typically do on naked, non-modified peptides. And then there is the step of mass spectrometry, and I think it's fair to say that in the field of PTMs, the standard mass spectrometry most people work with is label-free quantification using data-dependent acquisition. However, there are interesting alternatives, what people also start doing a lot at the moment, and that's the reason the IA's SWATH technique, that's why you're all here. I just also would like to mention there are also other interesting methods that people try out, especially also in the field of PTM, like TMT-based quantification and using the fusion lumos to do MS3 and synchronous precursor selection stuff, and which I will not talk about today. We will only just focus on SWATH, but there are also, of course, other interesting mass spec things ongoing. And now the last step, data analysis. And here I would like to summarize what I think are the major challenges when we work with DDA data and PTM. And many of these challenges have, of course, been named already. So one big issue when it comes to PTM data is missing values and limited reproducibility. And again, this issue, which we heard already many times before when we were talking about full proteomes or protein concentration measurements, this issue becomes even more severe when you do PTM analysis, because when you want to do PTM analysis, you do not have the freedom to choose between various different peptides that all can represent your protein, but you're unfortunately bound to the peptide level. There is just this one sequence, and you have not the freedom to merge between different analytes. Then there is the issue of sensitivity. So PTM peptides tend to be of lower concentration than naked peptides, because not all PTMs are occurring to 100% on the protein, maybe just to 1%. So they all tend to be rather low, and also PTM peptides might be bad MS flyers, so they just might give a lower signal in the mass spectrometer. And that's all why the reasons why PTMs are often very much low in the noise, and you want to really be very sensitive to find them. And then there is this issue of PTM site localization. That's an issue that actually might not be an issue for some types of PTM. So sometimes, depending on the modified amino acids, it's actually very clear in a peptide where the PTM sits. But for other PTMs, it can be very, very difficult. And the typical example you've also seen many times before is phosphorylation. Phosphorylation can occur on serine, threonine, tyrosine most times. And unfortunately, we often have many serines, threonines, and tyrosines in the peptide, so it's hard to localize this. And I personally also think that this issue of PTM site localization also contributes to the issue of missing value that we see up here, because when the tools are not able to reproducibly localize sites over many samples and sometimes switch the sites, this also all leads to even more missing values and all these issues that we see above. Okay, and then there is a fourth challenge, and that comes from this bottom-up approach, because as long as we perform this proteolytic digestion for our analysis, we unfortunately completely lose the information about PTM sites on different peptides on the same protein. So if you have a protein that carries a modification at different peptides. In the moment when you digest it, the information gets completely lost if you have various different proteoforms present in your sample where the peptide came from. So as long as we digest, we cannot really do proteome form analysis. And the reason the way to solve that is to measure the complete proteins and to do top-down or middle-down proteomics. Now, with methods like SRM, PRM, and DIA-SWATH, I think we can improve on these first three challenges. And yes, missing values and sensitivity, we already discussed a lot throughout the week, why we can improve this and how fragment ion chromatogram information can be very helpful. And now for the second part, I would like to therefore just focus on this PTM site localization and how DIA-SWATH data or SRM or PRM data, fragment ion chromatogram data can help with this challenge. Are there any more general questions to PTMs and PTM workflows in DDA? No? Okay. So then let's have a look at the challenge of PTM site localization. And I thought I do this based on a very concrete example from my own research. That was a real-life study that I performed where I was interested in phosphorylation changes in yeast upon osmotic shock. So you see, osmotic shock was a very, very well-liked project in our group. A lot of people did osmotic shock in yeast. And what I did at that time is I measured 3 replicates of yeast and, before the shock and 3 after an osmotic shock where salt was added to the solvent. And then I performed what was that day the standard proteomic workflow in our group. I extracted the protein. I used TCA sampling to immediately kill all yeast enzyme activity. I worked with 8 molar urea in my lysis buffer. I digested the extracted proteins with trypsin, performed the titanium dioxide enrichment, and measured everything by DDA. And then performed label-free quantification. And what I got in the end was peptide, phosphopeptide results table where I got now measured intensities before and after the osmotic shock. And this is now taken from the original table, 2 example proteins. These were 2 isoforms, glycerol 3-phosphate dehydrogenase 2 and 1. And these 2 proteins appeared very interesting to us from this DDA analysis because for both of these proteins I found one peptide which occurred many, many times in my list. This first peptide here for GPT2 had 3 possibly phosphorylatable amino acids, namely 3 serine residues, and I found all of these 3 serine residues phosphorylated, plus I found 2 doubly phosphorylated forms. And for GPT1, the situation was even more complicated. There I had 4 possibly phosphorylatable serines, 5 possibly phosphorylated, and 4 of the forms I actually could identify and quantify, and also 1 doubly phosphorylated form. And what this table showed me was, first of all, okay, I have a lot of missing values, the problem we all know, but what this table also showed me that these different isoforms that I found seemed to be differently regulated. So some were downregulated, others were upregulated, and maybe some were not regulated at all. And when I saw this data, I thought, wow, is this now all really true? So are all of these different forms really in my sample, and are they then really differently regulated by the cell? Does it make sense, and how complex is that? And in order to address this question, of course, one thing that I could have done at the time would have been to use site localization probability scores, scoring algorithm like A-score, Lucifer, Andromeda, Phosphora S. There are many tools out there that you can use that give these different isoforms certain localization probabilities, and this can maybe help you to kick out some forms which are more likely and less likely. Nevertheless, all of those tools would just give you probabilities, right, of what is right or what is wrong. So what we decided doing was to perform targeted phosphoproteomics on those sites by, using synthetic reference peptides to get really a very good ground truth knowledge about what's going on. So what I did is I went shopping, and I shopped crude C-terminally labeled phosphopeptides of all the 3 different singly phosphorylated forms of GPD2 and GPD1. And then I started looking into the Y and B ion series that are actually formed by these different isoforms. And what I realized is that actually these forms are sharing a lot of Y and B ions, okay? So all of them are, of course, sharing everything from the C-terminus up to the first serine residue, because it's always the same sequence, right? And there were actually just these fragment ions shown here in green that were unique for either this phosphoryl form or this phosphor form. Okay, so now you can of course think, wow, that means this phosphor form here in the middle I can never detect, right? If there's not a single unique ion that describes that. However, that would be true if we would measure with our methods only a single fragment ion per peptide. If we would do an SRM measurement with just a single transition, yes, then I would not be able to ever differentiate or clearly identify this form from the others. But we are always measuring several fragment ions. And what you can do is you can find a combination of transitions that is unique for this and not for the others. And this is what we call, so for example, Y9 and Y8, this combination is just present in this peptide, not in this and not in this. And this combination of fragment ions that uniquely identify one form from the other is what we call a unique ion signature. Okay, and so unique ion signature fragment ions, which can be sometimes just one transition, but can be also several, will help you to correctly identify and localize phosphorylation sites in that case. Okay, so and what I did now is I simply started measuring these synthetic peptides that I purchased. And I did this with SRM, but at that time, but you can of course do it also with PRM, or you can do it with DIA SWATH in exactly the same way. And here you see the chromatograms that I obtained with the 3 peptides each measured individually. And then I put all 3 of them together and I measured the mix. And what you see is that all of these 3 different isoforms were actually almost baseline separable from each other. So what I learned from these initial experiments was all phosphopeptide forms are separable in retention time, at least on the gradient and the conditions that I used. And each phosphopeptide has a distinct pattern. So the fragment ion distribution looks all different for the 3 different forms. Okay, let's have a look at the more complicated case of GPD2, where I had these 5 different forms. Again, when I measured them individually, they were all clearly nicely there and separable. Of course, when I mixed all 5 of them together, it looked a lot more complicated. But still, when you dig into the data, I could see that there were just 2 phosphopeptides, namely the serine 2425. They were perfectly coeluting, so their signal was really just a sum up of both, while the other 3 forms were overlapping, strongly overlapping, but you could identify the different shoulders. So, very complicated case, 4 serine residues directly next to each other, 1 a little bit further apart. They were all having their distinct fragmentation pattern, and even in retention time, they were somewhat separable. Just this serine 2425 was completely coeluting. So now you can, of course, ask the question, let's first of all make the statement that if you have a case like this where you have strong overlap or we have even perfect coelution, if you do MS1-based quantification like you do with DDA and sugar proteomics, it will be impossible for you to differentiate these 2 different forms from each other, because when you quantify, you will just have the precursor intensity, and this will be exactly the same for all these positional phosphopeptide isomers. So with MS1-based quantification, there will be no way to somehow differentiate these forms. However, if you work with MS2-based quantification, SRM, PRM, or SWATH, it does become possible to differentiate perfectly coeluting forms by just focusing on the fragment ions that are unique for one form or for the other. So in my case, for example, if we would like to be able to discriminate this serine 25 from 24, which is perfectly coeluting, we need to look at the discriminating fragment ion between the 2, which was only Y5 in my case. And when you look at this fragment ion Y5, you see it's only occurring in these first 3 forms. It's absent in the other 2 forms. And in the mix, you can see this triplet of those 3 forms. And if we now focus on the 621, the fragment ion that is present in the second two isoforms, you can see now that these two isoforms are actually nicely separable here from each other. So I could use this Y621, Y5 fragment ion to actually really correctly, accurately quantify this 725 and this 727 from all the others. Okay? Any questions to that? So this is all currently done on synthetic peptides. So we're just talking about the synthetic, very nice, very pure. This is why this looks so beautiful here. Yeah. But we come to this also in a second. Okay. So to just summarize what I said once more, it actually retention time information. If you want to localize phosphosites or PTM sites in general, retention time information is really important because when you have a peptide that has several possibly phosphorylatable forms, it really matters whether these are co-eluting or whether they are separable. If they are separable in retention time, the task for you is to identify these different groups, and then you want to localize which of these peak groups is which isoform. And for this, you should use the unique ion signatures that I just explained to you. And once you have identified them, you can actually use, again, all the fragment ions to quantify them, and it will be a correct quantification. Okay, so separable in retention time, use all fragment ions for detection and quantification, use the site-determining fragment ions to say which of the peak groups is which form. In the moment when peaks are co-eluting, the situation is different. So then you just get this one peak, and now you can, of course, also use all fragment ions to say how many peak groups, to identify the peak groups, and are there many peak groups. But if you now want to correctly quantify, you can also, for quantification, just use the fragment ions that are unique from the unique ion signature. Does it make sense? Okay. So that's the first lesson that I learned from this work with these synthetic phosphopeptides. Accurate retention time information and knowledge about whether peaks are co-eluting or whether they are separable is actually really important for correct site localization, but also then to choose the correct fragment ions for quantification. And then there was also a second lesson that I learned from these synthetic phosphopeptide, and that regards phosphate losses. Okay, so this list that I showed you in the beginning is the Y and B ion series where the phosphate group always remained attached to the phosphopeptide. However, people who have maybe worked a little bit with phosphoproteomics know this very well. If we look at the fragmentation spectrum of a phosphopeptide, very often high intense peaks are ions where also the phosphate has lost, was cleaved off, so left the fragment ion. So two fragmentations happened on the peptide, one in the peptide backbone and one on the side chain. This is also shown here once more. So a typical fragment ion you often see is loss of -98 Dalton. And that happens when this bond between this carbon and this oxygen also breaks, and then you, H3PO4 leaves the molecule and you remain with a dihydroalanine. And initially I thought, well, these are all high intense ions, it's really great. I can use them. If I find a dihydroalanine, I know that was the position of my phosphorylation. However, there's one problem with that, because if you have a normal serine, not phosphorylated, it can also form a dihydroalanine by losing a water molecule. And that's also something that happens actually to a much less extent than when it was phosphorylated, but it's happening also to a significant number of peptides. So just to give you one example to make this once more very clear. So if you think you have this peptide phosphorylated at that serine present in the sample, and you use the Y7 fragment with a phosphate loss here, the mass would be of this fragment ion with the dihydroalanine would be 785.51. And then if you find this fragment ion and say, oh, I'm pretty sure I have this phosphopeptide in my sample, I localized it, this can be wrong, because the same sequence phosphorylated on that serine will have the same precursor, and it can also form the same fragment ion by losing a water instead of losing a phosphate. Okay, so the second lesson that I learned from these synthetic phosphopeptides is phosphate loss fragment ions are not good proofs for site localization, because I saw with my synthetic phosphopeptides a lot of these water loss occurring also on my synthetic peptides. Okay, so and just to wrap up this story and to come to some real biology, I just want to show you on 2 slides that going through this hassle of ordering the synthetic peptide, measuring all of them by SRM, finding out about these unique Eigen signatures was worth the effort for our project, because what we did now is we used a phosphor-enriched total yeast cell lysate, and we spiked these synthetic isotope-labeled phosphopeptides inside. That's the beauty of having them labeled right away. You can spike them inside. And in this crude synthetic form, it actually doesn't matter so much in money, whether they are light or whether they're heavy. So always consider buying heavy ones if it's just a few euro more that you pay. And this is the data, how it looked like for the GPD2 protein. So in blue, you see the 3 peaks of the synthetic peptides that I spiked in, and in red, you see the light signal. And from these experiments, it was now very clear that I have only these 2, serine 72 and 75 present in my sample. And because they were retention time separable, I could use the whole transitions. I didn't need to focus only on the unique eigensignature peptides, and I could quantify them before and after osmotic shock, and we found a slight but significant downregulation. And the same for GPD1. So again, in blue, you see this overlap of peaks from the synthetic peptides, and in my endogenous phosphor-enriched yeast sample, I only had 2 peaks that corresponded to serine 24 and 27. And there were only 2 peaks, and they were baseline separable. I could use all transitions for quantification as well. And then these 2 sites were also followed up on point mutation experiments in years later on, and then also those validated these results, and that we could really prove that we were looking at the right phosphopeptides. Yeah, we just discussed this yesterday also a little bit. So in theory, to see light contamination in the heavy is very, very, very unlikely, because it is very unlikely that every single atom will be light. I mean, we always, when we work with these silac peptides, right, we have 6 or 7 or 8 C atoms or N atoms that carry it. And it's more likely, of course, that one of them might be light impure, or 2 of them. So you get a distribution, and to have the completely light version is very, very unlikely in theory. However, again, as Mike said, you're not sure if what the manufacturer said, that they worked with 99% purity, if this is really true. And that's why you should check, and you could see it. And I think, it's even just if the technician in the lab, wasn't working 100% pure, and they had some light impurities, or not completely cleaned the vessel, or this can also be all contamination, why this is not 100% correct. So it's always, always, always good to check that you don't have light impurities that you spike in. But you can check this very easily by just measuring your peptide light and heavy in water. And you can also prevent this a lot by not spiking in 1,000-fold more of your heavy than what you measure than in your light. But it's not more likely to have light impurity in these crude peptides than in the heavy. So crude peptides is actually often exactly the same workflow what the company does. So it's not completely different. It's all FMOC-based peptide synthesis. And they all start just with a building block that carries the heavy lysine or arginine. So in terms of impurity with light, this doesn't mean crude is worse or bad. Crude just means the product of the synthesis was not purified. Okay, so in the last part, I would now just like to give you some more specific considerations about what's important to consider about PTM analysis and DIA SWATH now, because so far all the data that I showed you were SRM data, even though, and that's what I want to say, to show you this actually doesn't matter. The concepts are still very, very similar. So how can you use all of that I told you for your DIA SWATH work? And the first answer to this is, if you are interested in a small-scale DIA SWATH analysis, so if your project is not measuring thousands of peptides, but you have a specific question, you can do exactly what I did. Get synthetic reference peptides ideally, and measure those, not with SRM as I did it here on a TSQ Vantage instrument, but measure them in PRM mode or in DIA SWATH mode on your instruments. So you would also just generate a PTM-specific library from your PTM reference molecules that you have. You would ideally then spike these PTM peptides into your sample if you have them heavy labeled, perform DIA-SWATH measurements, extract first the most intense transitions to be able to really sensitively detect all P groups that are present. And once you have detected your P groups, you will focus on the site-determining unique eigensignature fragments to make sense of whether you localized correctly. And Skyline is a fantastic tool to use for exactly this purpose and play around with the data and visualize it. And then you perform your quantification based on what you see. Do you have separation or not, and everything that I just explained you. Now, when you look at the details, there are actually a few things that would be different if you would do DIA-SWATH compared to SRM. First thing that is different is you have larger precursor isolation windows when you do DIA-SWATH compared to what I did with my SRM measurements. And larger precursor isolation windows means always more P groups of co-eluting fragment ions. You always can, so you can end up with horrible pictures like this, also George will show it later on, where you have for a certain, where you see 5 or 6 or 7 or 10 P groups, and for you it's very hard to make sense out of them then. And there are two explanations why you see so much more P groups with DIA-SWATH compared to PRM or SRM. And the first explanation is because you have more possible interferences, right? And that's what's always not so cool, and that's always what makes DIA-SWATH more challenging, because it might just be that by chance there are peptides with a similar precursor and similar fragmentation. However, when it comes to PTMs, there is also a second explanation why you have so many P groups here, and this is that there are other modified forms of your peptide that have a modification that is small enough that the precursor mass is still within the isolated window, and because then both modified forms are isolated and fragmented together, you end up seeing these many different P groups. And that's actually a very, this can be a super cool biological finding, right? This can be biologically super cool for you. This is not bad, it's just an opportunity, and it can be very interesting to learn about other PTM forms in your data. And that's something that you would not see necessarily with SRM or PRM. Okay, and when you work with C-terminally labeled peptides, as I did, this Silac type of peptides, one thing is also very important to consider is which ions can you use in the DIA-SWATH analysis, because we have here exactly the same problem. All the B ions with peptides that are labeled at the C-terminus will not carry the heavy label. So if you isolate a heavy and a light peptide and DIA-SWATH in the same window, the B ions will be exactly the same for heavy and light. So that's a small technical thing that many people stumbled over in the beginning. When you do DIA-SWATH and you work with labels, make sure that if you co-isolate heavy and light, you can still distinguish the two forms. People look a little bit puzzled. Is it clear? Yeah? Okay, and then there is the third thing that is also a bit different from DIA or PRM data compared to SRM data, and that is we have MS1 information. And I think also Alexei stressed this a lot already. You have this additional MS1 information, and this additional MS1 information, again, in the context of PTMs, can be very, very, very useful for you, and you can use it. Because if you have a case like this one up here, it might be that one is the methionine oxidized form, one is the phosphosite on X and phosphosite on the Y serine. So different forms that have a different precursor, you will be able to differentiate by looking at coelution from MS1 and MS2. So by taking into account MS1 information, it might be possible to differentiate which of these P groups that you see on MS2 level actually belongs to one or the other isoform. So MS1 information with PTM peptides can be very useful and make use of it. And most of the tools that we use for analyzing PTM data in an automated way do this also. Of course, it will not be helpful for differentiating phosphopeptide isoforms because they have exactly the same precursor mass. So there it will not be helpful, but differentiating PTM forms that have a different precursor mass, it can be very helpful. Or differentiating a PTM form from the naked form, for example. Okay, so that's what you would do if you would be, as I was in the past, small-scale studies. Now, what most people want to do, why they use DIA-SWATH actually is because you can go large scale and you can analyze thousands of PTM peptides. And then, of course, what you need is an algorithm that does the scoring for you. You cannot manually go through these data. You cannot order all synthetic isoforms for every peptide and all of that. So we need smart algorithms that do that. And there are actually 4 different ways how you can automatically analyze PTM SWATH data, and George will tell you much more about it, so I just very briefly would like to mention this. So there are ways and algorithms out there where you can perform open modification analysis of PTM data. That means open means you do not need to specify in advance which PTMs you're interested in. The algorithm just tries to find PTM carrying peptides, and you can do this in a peptide-centric or spectrum-centric way, as we have heard many times throughout the week. And there are also option 3 and 4 where you perform closed modifications. So there are also these projects where you know exactly which PTM you're interested in because you have done a phospho enrichment. So of course you want to look for phosphopeptides only or some other form of enrichment. And again, also here there are peptide-centric and spectrum-centric tools that can do that. And at this point, I just would like to highlight which of these tools are. And again, George will tell you much more about it. So these inference of peptidoforms, this IPF algorithm is something that George published last year. And this is an algorithm that performs closed modification. So you say that you're interested in PTMs usually, in which PTM, and a peptide-centric workflow. And this IPF actually works very, very similar, or uses the same lessons that I just showed you before. So this IPF method also first looks at or defines detection transitions, the most intense transitions to define P groups, and then it looks at the identification transitions, the unique ion signatures, to make sense and to localize the PTM site. So this algorithm really uses the concept that we learn from these SRM studies and put it into a code. And George will tell you how this works in detail. Yeah. But there are also other algorithms out there. A similar slide was on already. So there's this PICT algorithm. That's a spectrum-centric closed modification tool that uses DI-Empire and Skyline and MapDIA. So another workflow to achieve exactly this site localization of PTM carrying peptides. And this SWATH Profit PTM is an example for a peptide-centric open modification algorithm. So here you do not need to specify if you're interested in phospho or ubiquitin-related peptides. But what this algorithm does, it uses information from the naked peptides, so the peptides, the non-modified peptide it has in the library, and then it looks at the lower ranking P groups that it finds and tries to associate a modified peptide to these lower ranking P groups. And yeah, without having specified which PTMs you're looking at, it can give you a table of PTMs that it found in the data. So where do we stand with PTMs and DIA? So in the past years, several papers, a lot of papers, and here just examples, have been published that tackled the study of PTMs using DIA SWATH, phosphorylation, histone modifications, stoichiometries of lysine acetylation and succinylation were studied, and glycosylations were studied. However, there are also a lot of PTMs that have so far never been tackled with DIA, and there is a lot to explore, I think, and to do. So on this slide, I just try to summarize, so where do we go? What will be the future? And I guess what for sure will be the future is that our mass spectrometers will further improve and being faster, better, more accurate, more sensitive, and this will of course all have an impact on the PTM data quality and will make our measurements more sensitive and hopefully more accurate. This is for sure going to happen. There will also for sure be more improvements in the software tools. We will have hopefully more public PTM libraries or very robust workflows for library-free analysis. And these improvements, I think, will all lead to the fact that DIA SWATH analysis will be more easily applicable for many people, not just specialized labs or not just for people like you who took a whole week of a course to be able to run these tools. But I think next to these improvements which will come, there are also a few unsolved challenges, or I call them the holy grails in PTM research, which are maybe not so clear or not where people so far maybe they're not so many great ideas how to solve these issues. And one is, for example, the development of PTM detection strategies. There's still a lot to be done. It's not DIA specific, but there are just many PTMs which we cannot detect because we cannot enrich for them or because they're not stable under the conditions of our mass spectrometers. And I think that will be necessary to develop new ideas there. Then top-down, middle-down proteomics. That's something that we really need to do if we ever want to be able to differentiate proteome forms. And again, that is something that is, I think, still pretty challenging. And a last challenge, which I personally also find very, very important, is that we are currently not so bad in detecting PTM sites and making long lists, but what we're really lacking is assigning biological function to these long lists. We can produce a list, but we have no clue what these PTM sites are doing. And therefore, I think what's really very urgent as well is to develop smart ideas how we can put biological meaning to these long lists. And there, my personal hope is that with high quality DIS data, it might become easier or better possible to perform correlation analysis with other orthogonal data and do this type of analysis. Okay, with this I have some take-home messages. Okay, Ben? I quickly summarize what I think are the most important take-home messages from this lecture. So the success of any PTM project will always depend on the workflow. So, and we have all these steps, they are important. So if you plan a PTM and a project, really make sure you already start with a good extraction protocol, spend a bit more time thinking about this than maybe with just quantifying protein concentrations. A particular challenge that we have in proteomics is PTM site localization. And from working with these positional phosphopeptide isomers, we learned that site-determining fragment ions can be used to localize PTM sites, and that retention time information about whether things are coeluting or separation is really, really important to query the data in the right way. And coeluting or overlapping peaks can still be accurately quantified if you work with site-determining fragment ions, and this is something you can just retrieve from MS2 data, from chromatographic MS2 data, and not when working with DDA and MS1 data. And phosphate loss fragment ions are really not suited to confidently localize a site. And then some DIA-specific considerations. Problem of DIA, we have wider precursor isolation windows, which means more interferences, which is a challenge, but it also means you can possibly detect other unanticipated forms or other PTM forms, which can be very, very biologically interesting, and therefore it's a great opportunity. And there are exciting times ahead because, yes, these algorithms to make sense out of such data are just developed. These papers are all very recent, and they are still under development. And yeah, automated PTM and site localization of DIA data, I think, yeah, is in its, it's just making momentum and speeding up. And George will tell you a lot more about these PTM software tools later on. Thank you very much for your attention. Thanks a lot to these people for contributing slides. And I'm happy to hear any more questions.